this time I would like to use computational chemistry to explore some questions that current laboratory experimental studies can't exactly show so easily. So I selected the Hydrolethalus Syndrome because this is I think, an appropriate situation where the current techniques probably lend itself only to the realm of computational studies. So basically, I'll talk about first what Hydrolethalus Syndrome is. So Hydrolethalus Syndrome is a very debilitating condition. It was first discovered about 40 years ago in Finland, and it was discovered by accident during a nationwide study on Meckel syndrome. It turns out that Hydrolethalus syndrome leads to severe nervous system malformation, and as you can see in this still baby here, this still fetus, there's encephaly, which is missing parts of the brain and head. There's occipital bone defects. There's also respiratory tract issues and congenital heart disease called truncus communis. There's also broad neck that's characteristic to Hydrolethalus syndrome, cleft lip, and palate. Now, the incidence of Hydrolethalus syndrome is kind of rare, but somehow it exists mostly in the Finnish population. A few studies were actually shown a few cases that were actually found outside of Finland, but this is primarily localized to the Finnish population. Also, you can see polydictyly and duplicated too. So these are key features of Hydrolethalus syndrome. So the genetic profile, Hydrolethalus syndrome is actually a missense mutation that takes place on chromosome 11. And chromosome 11, interestingly, is a chromosome that is responsible for a lot of diseases, including autism, from autism to bladder cancer. So what's happening with Hydrolethalus syndrome? Well, in simple, there's an amino acid mutation. There's one simple mutation from aspartic acid to glycine, and that takes place here on the Q24P locus on chromosome 11. So there's one non-conservative mutation. We know glycine is a negatively charged amino acid. Aspartic acid is a negatively charged amino acid, and glycine is a neutral amino acid. So we consider it non-conservative. But interestingly, this mutation takes place in a region on the DNA that is highly conserved across species. So from C. elegans, from little tadpoles and insects, all the way up to humans, this region of DNA cannot tolerate mutations easily. So the basic message from this slide is that this amino acid has some critical function, and this is why I wanted to use computational chemistry to explore the importance of this mutation. Now, let's talk about ab initio structure. Ab initio simply means folding from scratch without any templates. This is used to predict three-dimensional structures of proteins, and interestingly, predicting three-dimensional structures from scratch is one of the 125 outstanding issues in modern science. So today we understand it pretty well, but we still don't have the full picture about how to fold proteins, how the body folds proteins, how nature folds proteins. If you take a look at this image on the right, this protein is naturally folding by itself. It has, based on its amino acid sequence, it's folding up into a particular structure that is related to its function. So today there are limited abilities to fold proteins by ab initio methods because most methods for protein structure prediction rely on homology. Homology means using other solved structures experimentally to predict a structure. And you can look at protein families, and based on your family, you can say, well, you will have these characteristic features. So one of the limitations with ab initio protein modeling is lack of force fields. We basically employ physics and thermodynamics to fold these proteins according to nature, how nature would do it. And the biggest challenge is to fold the protein to what is called its native state, and that's the energy minimum. So the protein on the right has folded up properly. You can see there's an alpha helix that's spirally here, and maybe one sheet here, one beta sheet in this topology. So for this study, I used two supercomputer servers. The first is called Robetta, and this is spearheaded by Dr. David Baker at the University of Washington. And in fact, this is a world-renowned server that you can input for simulations on protein folding and molecular dynamics. The next server that I also used to compare the methods is Hydrolethalus, and Hydrolethalus is developed by Professor Zhang at the University of Michigan Medical School. So I used two servers independently to see if the results will make sense. So here's an example of comparative modeling versus A/B initial. In comparative modeling, we can take proteins that have families, for instance, hemoglobin or myoglobin, and there's a lot of families. If we don't have the structure of rat hemoglobin, we can use humans and model it of human beings, So A/B initial modeling is where there is no family templates, so you have to fold the protein from scratch. Now, we fold proteins based on force fields, and a system, according to the rules of thermodynamics, energy is not created nor destroyed, it just changes form, from one state to another. So when we look at a system in a protein, we consider it with total energy. And based on this, we can use we can break down the force fields into different components to fold these proteins properly. So the first layer of force fields we talk about is the atomic level force fields. And this basically look at how nitrogen interacts with hydrogen, how hydrogen interacts with oxygen. The second layer is what we call residue level. So how the amino acids interact with each other, how glycine interacts with aspartic acid, how arginine interacts with lysine. And then the third layer deals with the three-dimensional structure of the protein. We call it topology. How does the alpha helices and beta sheets relate to each other on a global perspective? Global meaning the entire protein itself. So these are the terms in specific. First is backbone. Proteins have backbones. They have side chains. They have excluded and included volumes. We see hydrogen bonding. We see also things like solvent accessibility. When a protein is in the blood is the solvent, right? Serum, and it has a particular pH. We look at blood backbone torsion potentials, how the protein is, how flexible it is. We look at distance profiles, radius of gyration, helix packing, and strand packing. And based on this, we can then model the protein in fragments. And then the last challenge is to identify a minimum energy. And proteins like to have the lowest energy possible. And that's a challenge, I think the biggest challenge in A/B initial modeling. So the results of this study, basically after 2 months of modeling, so I computed about almost a month and a half of modeling, we ended up with this structure of HYLS on the left. And what is interesting about this predictive model of HYLS is that the servers and computational methods predicts that this protein has 61% of intrinsic disorder. So what is intrinsic disorder? Intrinsic disorder basically refers to regions of the protein that are not helices, that are not coils, and parts of the protein that are not beta strands. So this is like this area in sky blue, we consider it intrinsic disorder. Now the second characteristic is that 36% have well-defined structural elements. So basically we can say that the Hydrolethalus syndrome protein is highly intrinsically disordered. And this is a big problem. Why is this a big problem? Because we cannot use any experimental methods really currently, which is nuclear magnetic resonance spectroscopy and X-ray crystallography. Those are the two main techniques to visualize proteins. We can't use them because this protein is intrinsically disordered. It's very, very flexible. It's hard to characterize. So we must turn currently to maybe computational methods. this is what I basically found. I found my conclusions were there's a helix disruption hypothesis. And what is happening is the mutation, so this is the wild type protein, the normal one, and this is the one that causes Hydrolethalus syndrome. There's a mutation here from aspartic acid to glycine. And notice that when the wild type, the normal protein, there's good interaction between arginine and aspartic acid. Here, hydrogen bonding and electrostatic interaction. But when we introduce the mutation, there is no more hydrogen bonding. So what happens is we call it the helix disruption postulate. I believe that's taking place. So we're actually breaking down the helix and forming a coil. And breaking down a helix and forming a coil is what we call misfolding. So here the helix misfolds, and here the helix misfolds. So here's the nice blue helix here in the wild type or normal protein, and in the mutant, notice that the helix unfolds here. Helix unfolds completely. This, I believe, exposes what is called the hydrophobic regions of the protein, and this creates a sticky patch. So proteins start to stick to things, stick to other proteins, stick to lipids like fats in the body, and this creates a big problem. So there was a study done by this. There's a study published in 2005 that studied the mutation Hydrolethalus syndrome in mouse models. They started with cells and then they went on to mouse studies, and what they noticed was that in the wild type, in the normal expression of the protein, the Hydrolethalus syndrome protein doesn't exist in the nucleus. But in the mutant form, we notice that the protein can enter the nucleus. Not only does it enter the nucleus, but it forms what is called inclusion bodies. You see these hyper-intense dots here? They're inclusion bodies. So these are protein aggregates that stick to each other. Another example would be in Alzheimer's disease. Prions stick to each other, and that deteriorates the brain. So the group further, in the further study in 2005, this group also showed that this protein during mouse development, during neonatal mouse development, this protein is expressed in many regions in the brain and in the developing organs. So no wonder if you mutate this protein, it would affect the entire fetus and the entire, and that would create major problems. So in conclusion, inclusion bodies are hallmarks of genetic diseases like Parkinson's and Hydrolethalus syndrome, and even in albinism, right? They are hallmarks of many diseases and disorders. The second is that intrinsic disorder proteins are actually challenging to characterize. In the future, there will be methods to solve this problem, but currently, it's a big challenge. And so we need to turn to now computational studies to actually probe into the etiology of HYLS1 and why this mutation causes this lethal condition. The next conclusion is that the results actually provide a framework for the development of druggable proteins, protein-protein interactions, and that might have major implications in Alzheimer's disease. There's a professor from Cambridge who passed away 2 weeks ago, a very famous professor, and he at Cambridge University, and he showed, Professor Chris, and he showed that, he demonstrated that protein-protein interactions can actually be inhibited. And if inclusion bodies are protein-protein interactions, then we can use that to actually cure maybe perhaps Alzheimer's disease. So I'd like to thank Isho University, my professors, my friends and family, and thank you.